I want to showcase the methods, the technology advances we have made, and in the field that with really exciting results recently we have gained. So HIV infection is one of the projects working in the lab. So the life cycle goes on. The HIV, starts off as an immature virus. Then it has immature viruses not infectious. It has to go through maturation process and converts this spherical particle into this conical shaped core that contains genome. Only this one's mature. Only this mature virus is infectious. Now, when the infectious virus gets contact with the target cell, the GP120 and the surface receptor bind together and the membrane fuses, release this conical shaped core that again contains genome. The whole core has to go through the entire cytoplasm and get into the nuclear pore, into the nucleus. And this process is only being, found there until 3 years ago. We all think this whole thing capsid has to come apart in the cytosol, release the genome, and only the pre-integration complex gets into the nucleus. But this is all new now to us. Now we want to understand how this happens and what involves in this process. There are many host factors that associate with this process. And there are restriction factors in red, trim fiber, MXV, and those dependency factors that require for infection shown in green here. We study those factors in complex with capsid as it going through the cell. But today I cannot talk about all those structures of those, but focus on two areas. One is the structure assembly of immature and mature particle, and a small molecule called IP6. The role of IP6 plays in the assembly of immature particle. And in the second half of my talk, I'm going to talk about the nuclear import. This is a brand new area. It's just so fascinating now. We found that the whole entire capsid can go through the nuclear pore. So when I started working with HIV capsid assembly, we use this in vitro system, recombinant protein induced assembly. They assemble with a long helical tubules with hexamers on them. Very few pentamer, but mostly hexamers. Actually, Tammy here also worked on this. But they assemble in a very non-physiological condition under 1 molar salt and a pH 8, which is unthinkable in the cell, right? So we didn't know what was missing until much later. I mentioned IP6. We were missing very important ingredient called IP6. Nonetheless, back then we didn't know. So we used this in vitro assembled, just like a crystallization traces or in vitro in unphysiological conditions. And then we determined the structure to high resolution. What we found is the hexamer actually here, unlike the crystallized hexamer and anything else, each hexamer is curved. They curve in a different way. So basically, each hexamer in the conical tube here, every hexamer is different, not the same. Each tube hexamer, they have a different curvature, and the hexamer curves differently. So this is one of the example here. So if you cut across this slice along the axis, near axis of the tube, you see hexamer on this direction is flat. And if you cut across this direction, the curved direction, you see hexamers all curved. And each diameter here, the assembly is very heterogeneous. The different diameter actually reflected in the conical shaped core, and the different diameter has a different shape and a different curvature of a hexamer. So that's what we found. Actually, it's relevant to it. We study also relevant. The curvature is relevant to the host cell binding, for instance, cyclophilin binding, and also relevant to the small molecule. Now, previously called PF74. Now, this is lenacapavir, which is a very good drug now, important drug for HIV treatment. And now it's FDA approved. You only need to take every 6 months in one injection for antiviral treatment. So, but this is as I said, this is non-physiological. What we want to really look at is a physiological condition, what capsid looks like. So we want to into entire particle. And this was also previously done in John's group by Simon Matty as well. So, but we found that these particles are quite images are very noisy because when capsid, immature capsid, GAG cleaves, it forms a conical capsid. Only half of the CA protein went to assembly, other half floating around in the virion. So that contributes lots of background. So we devised a method called performing protein. They lack cholesterol-containing membrane. We treat the virus with this PFO, and they poke holes on the viral membrane. You see, this is a pre-pore formation, the two protein bond. But when they changes conformation, that they make about 40 nanometer holes. And this holes allows all those soluble protein, the CA, unassembled CA leaks away, and they have a great improvement of contrast, so we can get a higher resolution structure on this. Not only this, we can also introduce many host factors go into bind the capsid. And for instance, this is a cyclophilin ADS red, you can see bound to the capsid, just go into the hole bound the capsid. And then we can determine the structure of this. Now we take this assembly, look at the native authentic capsid now, and when we look at structure, okay, we got the hexamers about 5.8 angstrom. Then we see two little densities here. Now, other work has done by Owen Pinellas and also Greg Towers and Leo James' work show that actually IP6 binds to hexamer. Now we see, they were crystallizing and crystal structures from. So now we see two blobs in the native authentic capsid. These blobs, we thought maybe this is IP6, but why is this only little? Because the IP6 also leaks out as in this, when we poked out. Now we add IP6 back, and then we certainly recover these two densities. So we know they are IP6, and then we work with the molecular dynamic simulation group that shows that the two IP6, there are two binding sites, two IP6. One binds the arginine 18, we all know. Actually, the second IP6 has that binds to lysine 25 and lysine 30. So this is their coordination. Now, where the IP6 come from? You know, this is in the virion. It has to be there. It comes, of course, coming from immature particle. Initially, it was packaged. So we look at how they immature particles package IP6, right? So so this is what they look like, and we made two different kind of preps for immature particle. One express this protein. Actually, we made a mutation called T8I that stabilizes immature assembly. In E. coli cell, they automatically assembles the immature particle in the bacterial cell already, and without membrane, of course. And we also make those T8A GAG VLPs, but also from HEC293 cell, and shown here. So those ones has membranes. And we determine the structure using tomography, subatomic averaging, and EM clarity that we developed. So shown here is a T8A GAG, and from this is E. coli, this is from VLP from mammalian cells, HEC293 cells, and then you see the structure is very much similar. But we can compare the early structure done by Florian Shaw in John Burke's group. Now, they have a wild type, but with maturation inhibitor BVM bond. And the GAG, actually the GAG, as we call the 6 helix bundle domain, SP1 domain, SP2, is a stop short at the T8. And the T2I mutation actually extends the 6 helix bundle all the way to the unseen nuclear capsid region. So this is why we found that the T8A actually stabilized the immature assembly, because it forms a really good 6 helix bundle. Now, when we look at the structure, it cut right in the middle of a GAG, we saw the density now. This density, actually, we made it in E. coli. We don't know what it is, but in the mammalian cell, this is IP6, we know. We still try to figure out in the E. coli cell, what does the GAG recruit? Because E. coli cell doesn't make IP6, doesn't have an enzyme for, they don't have an enzyme to make IP6. But this pocket, as you can see, the two lysine, the 6 of them, 6 lysine on top, 6 lysines in the bottom, is highly, highly charged and hunger for something to go in. So this is IP6 where IP6 is, and in E. coli, some polyion probably sit there. So we try to understand. So if we mutate those lysines, what happens? So we work with Eric Fried at the NCA Frederick. Now, he generated all those mutants, whether with a single mutation of lysine 2 to 7, double new mutation, and another one's a double mutation with a T8I mutant. And along this, with wild type, we saw IP6 density, but with single mutation, we don't see much of IP6, and with a double mutation, even with T8I, we see no IP6 density. Now, even without IP6 density, they still assemble the immature particle. So this indicates that assembly of immature particle really doesn't need IP6. The virus is doing all this just for the purpose of mature assembly. Eric also grew this virus and come up with a revertant mutant. And the revertant mutant was a G225R. Remember, this was a 227 here, right, lysine? So 225R, we thought, oh, no, the G mutant to R to compensate lysine. Maybe, no, this is G225 actually recovers the infectivity that allows to binding recruit more IP6 and helps the mature assembly. But in fact, we determine the structure. There's no IP6 in G225. The arginine is pointing away from the center channel, not instead of like a lysine pointing in. So now we actually, I don't have time to show data. We understand that G225 actually stabilize the mature assembly without IP6. This is a very interesting and a very good tool to use for structure and for other studies as well. So G225 recovers infectivity from those mutations, but recover in a way not to recruit more IP6, but stabilize mature assembly. This recruitment of IP6 in mature particle is actually very conserved, highly conserved in most of the retroviruses. We studied called a human endogenous virus called HERV-K, and we determined the structure and also saw this little density here with two lysine, also sequences different two lysines coordinating this little IP6. And again, Robert Dick also determined the EIAV structure. They also see coordination of IP6 with the charge residuals. So the story now is very interesting. So I say virus is really smart, like a chess player, and thinks about several steps ahead. When they buzz out from the cell, they use this high affinity coordination and to ensure that every hexamer has an IP6 in there when they bud it off. And when their protease cleaves the gag and forms a mature particle, I can calculate how many gags there and how many IP6 will be made, and that will be released. So there will be 1 millimolar IP6 in this intracellular less than 100 molar. So after cleavage, you will have 1 millimolar IP6. That ensures that during mature assembly, and this IP6 will be incorporated in the capsid to stable assemble the capsid. Now, when they goes back into the cell, again, exposed to less than 100 macromolar IP6, and then, the IP6 may lose, and then this central hexameric channel that allows the DNTP to go in, and this is required for reverse transcription to take place because you need a nucleotide to do reverse transcription. And this is a low affinity. So virus modulate this different coordination system for the same small molecule for different purpose. So very nice story. Now I'm gonna switch gear to talk about nuclear import. Okay, this is all new. Nuclear import is it's very rare and very, very transient. It's very hard to catch. So we have to develop a lot of ways to overcome this. The first one is we have to look inside the cell. So we use film milling, and this is a video taken from some official website. So this is a dual beam machine. Use a cryofib to trim the cell into a very thin lamella. Then we use tomography to collect T series and to reconstruct a 3D volume. And shown here is a segmented volume, I think by Julian Mohammed. You see the nucleopores acting filaments. Now we use this methods technology and to cut very, very thin lamella on the T cells, which is relevant to HIV infection. So we certainly can see when you cut the lamella extremely thin, about 80 to 100 nanometer, which is quite challenging to do, you suddenly see a lot of clear pictures in the cell. This is T cell, you see nuclear envelope, and this nucleus is cytoplasma, you see ribosomes everywhere, And you see chromatin fibers that has never been seen so clearly, so crisply before. You see fibers here, fibers there. You see individual nucleosomes. This is a zoom-in view. You see one fiber. And you see sometimes a short range of a nucleosome arranged into a higher order structure. It's better, shown better in this movie now, showing same area now. So those are ribosomes, those are cytosols. This is nucleus, nuclear envelope. And you see individual nucleosomes. Those are really small, 200 kilodalton stuff. We can see it in cell, in situ, in the native live cell. And we can pick out all those small particles and arrive at subatomic averaging structure, which is about 11, 12 angstrom resolution. You can see nucleosome structure there. Now, this is very challenging to push it further. We add more particles and couldn't get it even better. I believe the nucleosome there is highly dynamic, highly heterogeneous, and bonds with binding with many chromatin modification factors. So this is what we get. And we also can analyze those nucleosomes, whether they have a linker histone H1 there or not, and how they distribute. It's very important because this picture shows, how you package 2 meter DNA into 10 micron cell nucleus. There's models of DNA wrap around the histone octamer and form nucleosomes, and the nucleosome form secondary order structures that's called 30 nanometer fibers. Then they further organize a 3D genome in the nucleus. However, there's a prevailing models for how the 30 nanometer fibers are organized, either with solenoid model or with a zigzag model, although this is now more prevailing and more commonly accepted. But this is all based on in vitro study. No one has seen those, actually, what they look in the native authentic nucleus. Now, we see those nucleosomes beautifully, so we can we can see how they are connected together. So shown here are 2 fibers here, and we can look at individual each nucleosomes here and connect them because we know where the H1 histone is located. And we can link them together, and you can see this is one fiber here, one next fiber here. You see how the linker, we don't see linker DNA. Sometimes we do see, but not all of them. And you see how the nucleosomes are linked. And this is like a low range partial high order structure that has been kind of captured in vitro, but they are very rare. Most of them are actually very flexible, no rigid 30 nanometer fiber, and they form zigzag flexible chromatin fiber. But of course, this is one state of, this is from T cell. We yet to look at the other cells. We do look at the other cells. They, for instance, these cells are very difficult to see anything. And they also, we can look at the different stages, like this is the interface. You can look at the mitosis S phase or other G phase, G1, G2 phases. Now, of course, this is not our interest. This is a byproduct. When we look at the control T cells, we suddenly saw many new things. This is what is about discovery. You have a new method, new technology on the horizon, and you use it, and you suddenly discover new things. But we are really interested in HIV transport. So this is a cartoon model shows how the capsid gets imported into the nucleus, and there are NAP proteins that has FG motifs drag the capsid going through the nucleus. Now we've been working on this for a few years, and this is work done by Zheng and Stanley. Initially, we just brand milling it and try to find it at the nucleus. So we can find the nucleus and mill it, see if we can find the HIV core. And this has been very challenging. We mill 100 lamella, and in 100 lamella tomogram, we only find one capsid. So this is not the way to go. And we are not alone, actually. In a recent bioarchive paper put out by Martin Beck and Crosslink, and the paper, they also show they only capture total nuclear associated cores in their paper. So this is a common problem that we have. It's a rare event. They are rare events. They are transient, very difficult to catch. So we, to overcome this, we developed 2 advances. One is to establish an in situ or in vitro reconstituted system that we reconstitute a functional transport system to study the nuclear import. The way we do it is we permeabilize less T cells. So we remove the membrane, and then we add isolated core to it. So this way we can capture loads of cores go onto the nuclear envelope, the green one here, and co-localized with NUP in red. And you can see better. If you just look at the HIV, green label, integrase label HIV, you see just almost decorating the whole envelope. So we get loads of them at least. And sometimes we know it's functional because you see the two green dots inside the nucleus, they actually transport the HIV core into the nucleus. So that's one. Another one, it's a movie and showing this loads of envelope loads of HIV core as associated with the nuclear envelope. Now, second one, we developed a 3D correlative targeting for HIV cores. And this is precise. Okay. Quickly go through it. Okay, because this is very exciting. The most exciting part. So we can target the individual particles in the cellular lamella and to collect tomography data. Okay. So I show you now, this is the method I explained, and we can capture individual particles in different stages of transport, even in a single tomogram. Showing here that you see those particles are, this is a movie coming above, those are HIV cores you can see, and in the segmented volume you see, okay, here, they are in the different stage of a nuclear transport. Okay, some are approaching here. I wish I could speed up the movie. And some are actually docked on the nuclear pore. And this is just going through it, you see. And the other one is being just transported and in the vicinity of a nucleosome, you see. So we can do this, with these two systems we developed, we can capture hundreds of, over 500 nuclear HIV cores in the process of a nuclear transport. We can analyze how many are in different stages, and we can actually analyze what core goes in different stage. Actually, it turns out the smaller cores, only smaller cores passages through, and the smaller cores are actually being imported. And most of them being imported actually is the tubular-shaped core, not the conical-shaped core. We only found the two small conical-shaped core among those. And sometimes we see cores get squeezed, deformed as it goes through the nuclear pore. And so I'm not going to play the movie. And also we see nuclear pore actually gets modified and changed during when HIV inside. So you can see lots of nuclear pore with HIV capsid in here. Now 4 of them actually with occupied, and 1 actually is empty somewhere here. Or 1 is here empty. And we see the diameter changes as the HIV going through there. And also it cracks, sometimes the HIV core cracks nuclear pore, and I see the asymmetry of this. So I'm going to finish now with this beautiful image of a segmented tomogram showing that we can, in this advanced methodology state of art, we can capture HIV cores in a different stage of transport. And we also determine the structure of capsid associated with each stage of transport. They are different, as you can see already. So with this, I acknowledge all the people. This is my group in Oxford, and acknowledge people who did the work, and as well as collaborators, funding agencies. And thank you for listening. Thanks.
Thank you very much. This is a fascinating talk and fantastic work. And now this is open for questions. So, Tanmay.
Beautiful talk. Can I just ask about the IP6 parts of your talk? I was quite impressed how you resolve these densities. I think I have PTSD of Bivirimat-treated capsids. How did you manage to take care of these phantom densities that appear near the symmetry axis?
Yeah, we don't know what they are yet. I think some are probably real densities. Mm-hmm. We just don't know what they are.
So in the C1 also they appear? In
the C1 they also appear.
Okay.
Because C1 really is not C1, as you know. If the molecules are quite symmetric, And you really, even you do C1, you cannot differentiate the different orientation. If the density is not, resolution or is not clear enough, you cannot differentiate.
Just one quick one. Could the stuck capsids in the nuclear pore be a mechanism for disassembly?
It could be because there are models for, NAP358 may associate with the encoding, but also maybe encoding happens in the nucleus because CPS6 within the structure, they can disrupt, with low macromolar concentration, they can disrupt the capsid. So this is a whole new field we are going in now. It's very exciting.
So if I got it right, you've shown GFP labeled integrase, and can you use photobleaching to establish how many integrases you're looking at, or to count the number?
Yeah, we caught how many integrases in there, but we now have a better label. So this was done like maybe half a year ago. Now we have actually direct capsid label using non-natural amino acids with a click here chemistry. And the labels are much more uniform, and it also benefits labeling the capsid instead of integrase.
Okay, thank you.
So if there are no questions, we will move to the second talk.
Nothing online.
Nothing online. Okay, perfect. Well,
Thank you.
Peijun again.